Well, good day. What a remarkably erudite and engaged community you have gathered for conversation here in New Bern. I'm really impressed. Have already had so many good conversations. Not only a beautiful historical place, but great people. So delighted to be part of our conversation today. Well, there's a lot to cover, so let's go right into it. In 1975, there was a small group of scientists who met at Asilomar, a state park in California. They're all very busy people, but they took out time to meet together for a couple of concentrated days of conversation because they knew that they were part of just a few people in the world at that point that had discovered how to transform or change human DNA. They realized that all of life, all of physical life, uses the same basic DNA system. The recipe, the instructions for how to form a physical structure and how to run it, for how it survives. All of life, the same basic DNA structure. So now they had found a way. It was expensive, it was time-consuming, it was awkward, but they could affect DNA. The most concrete, immediate use that was found for this is one that might very well affect quite a number of you in this room right now. And that was if you have diabetes, if you inject insulin, it used to be the only insulin we could offer to you was porcine from slaughtered swine. Well, with this new technology, they could read the recipe for how the body makes human insulin. There's got to be a recipe somewhere. Your body has it. It's in your deoxyribonucleic genetic code. They read how the recipe reads, and then with their new technology, they inserted that recipe into single-cell bacteria and commandeered their machinery so that they started secreting human insulin, and then gathered that in sterile vats and distributed it. So if you use, for example, Eli Lilly's Humulin, that is a genetically modified bacterial source of human insulin that's never been in a human body, but can now function well as the insulin that your body's not producing if you're diabetic. All right? So that would be an example of a medication that was created through this new technology. Well, this handful of scientists who developed this capability were concerned, though, that now we had a way of directly changing this foundation of life, DNA. What if it was used to create a weapon? Or what if it was used to do this or that? It's so powerful. Could it be misused or abused? And so they met together and talked about how do we safeguard this new capability. And they actually came up with a set of guidelines. You can see a poster like this in almost any genetic lab, reminding people of what this small group devised together. That there had to be containment that was essential so that this wouldn't get loose outside the lab. That there would be biological barriers to limit spread. That there would be good microbiological practices and training. Basically, a whole set of guidelines to keep this technology safe. Well, the big change that followed not too much later, in 2020, the Nobel Prize Committee gave the Nobel Prize for Chemistry to Doudna and Charpentier, who discovered a naturally occurring gene editing tool. Now, here's the context. You probably all know that a virus is a package of DNA that injects itself into a bacteria cell or any cell in an animal body and commandeers the machinery to start making more viruses so that the cell fills up with thousands, tens of thousands of copies of the virus, explodes, and then they go out and infect the next cells. Okay? Well, bacteria over time developed a way to defend themselves from viruses. And what they did was bacteria developed a mechanism that we now call CRISPR, which could recognize the viral DNA that was foreign and chop it up into little pieces so it couldn't take over. Well, what Charpentier and Doudna did was they recognized this system, and then ingeniously they said, you know, if this can happen automatically in a bacteria cell, what if we could direct it? What if we could guide it so that it would make the changes and the cuts where we want it to, in precise points and places? And that's what they developed, and we now call CRISPR. So this is a technology which is quick, it's inexpensive, it's accurate, it doesn't go off places you don't want it to. It goes exactly where you tell it to. And it can cut DNA at a precise point, and then you can insert material or you can remove material. So an incredibly powerful technology, but relatively accessible. At Roanoke College, our students in their first course in genetics use CRISPR in lab. All right? So it's accessible, it's quick, it's inexpensive, it works really well. So what are the implications? Well, if general recombinant DNA was already a powerful technology, what about this? So in 2015, there was a joint meeting of about 200 geneticists and a handful of other people in other areas of expertise that were invited to meet at the National Academy of Sciences in Washington, D.C. for kind of a Silomar 2, to talk about, yes, we have this new technology. What do we need to do to be careful that it's used well? It's not enough to be able to do something. We need to be careful about choosing well what we do with this new technology. And so the group met for about 3 days. There were representatives from all over the world. It was officially sponsored by the Chinese Academy of Sciences, the Royal Society from the United Kingdom, the U.S. National Academy of Sciences, and the U.S. National Academy of Medicine. We met for 3 days, brought in food so that we could be very focused. Here's a picture of the National Academy of Sciences. Maybe you've been there. They're on the National Mall in Washington, D.C. It's right between the State Department and the main boulevard of the mall. There's a statue of Einstein lounging out front, if you've noticed that, if you've been on the National Mall. It's exquisite inside, done with marvelous detail of mosaic tiles, and amazing communication technology for being able to conference and work together among scientists. At this particular meeting, it was kicked off by David Baltimore, the president of Caltech, who's also a geneticist. And there were, as I said, representatives from all over the world. Pay no attention to that fellow in the upper right-hand corner. The global discussion since that first meeting has continued. The group met April 29th, 2016 in Paris, then again in 2016 in Washington, D.C., then in 2018 in Hong Kong, most recently just in March in London. Now, we don't have time to talk in detail about how CRISPR works. The quickest way you can get up to speed, I will mention to you, is that Jennifer Doudna, who won the Nobel Prize for this in chemistry in 2020, has done a very nice 25-minute piece that you can pick up on YouTube. And she walks through exactly how the process works and what the most current applications are. And she talks as the insider, the discoverer, about how CRISPR works. Now, by the way, when I use this word CRISPR, it literally stands for clustered, regularly interspaced, short palindromic repeats. So aren't you delighted? I'm delighted that we use this acronym CRISPR, all right? Much more easy to handle. The particular place where she describes how this works in a very nice and accessible way is called CRISPR and the Human Genome Editing: Progress and Opportunities. So I'll mention that resource to you. What we have time to focus on today is more along the lines of the philosophy, the ethics, not just what can we do, but what should we do. Okay, CRISPR, as I've been saying, is a key player for everything alive. These particular discussions focused specifically on human beings and how it applies to us. There were some key questions, and I'll just give you a quick example of what we ended up talking about. Should CRISPR be used only to heal a patient presenting for treatment, or should we also treat for their future children? Should gene editing only be used to cure disease, but not to enhance desired physical traits? What are desirable traits? Is it better to live longer? Would it be fair for some people to buy better genes than others can afford? Does editing someone's genes make them less of a person and more a product of our manufacture? Do we have a right or obligation to make such choices for our children and future generations? Theologically, is such playing God, or is it fulfilling a God-given calling? Who decides? The group often look like this, look rather pensive, right? I mean, people are really, there's lots of frowning, lots of concern. People are trying to figure out, whatever do we do with this? Well, the group did put together a communiqué, which was published at the end of the meeting, and very carefully worded. And I'll just briefly look at this with you to see what the group concluded. This was given out for immediate release on December 3rd. Incidentally, it was published in the London Times, the Wall Street Journal, the Beijing Times. It was published globally, and almost nobody noticed it. One of the main reasons for that was that there was I don't know if you remember this, on this particular date in 2015, there was a massacre in California where a civil servant went to the Christmas party for their division and shot it up, and then there was a police gun battle just outside. That, needless to say, dominated all the news that day. But very crucial, small piece talking about this occurred almost everywhere else. Okay, the text, I've just highlighted in gold some things I want you to notice. It starts out by saying scientific advances in molecular biology have produced remarkable progress in medicine. Remember, you probably don't worry about getting polio at this point. Although not that far back, people were very concerned about getting polio. Maybe some of you even had parents, or you remember that you were sent out of the city in the summer lest you get polio. We had a president of the United States who was crippled by polio. Polio was a constant threat, and now we generally just don't worry about it because we developed a vaccine to prevent it. So the scientists wanted to remind everybody right at the beginning, we've made remarkable progress in medicine. Keep that in mind as we think about what to do with this new technology. They also wanted to remind people that the scientific community has consistently recognized its responsibility, that the scientists themselves who are developing these techniques really want to do the right thing, and they're talking with each other and regulating each other, trying to make sure that these are used in a positive way. In particular, when we talk about CRISPR as a kind of genetic microsurgery, the most common term these days, because we're so used to it with software, is to call it gene editing, where just like a software program can go in and find a word, take out a word, add a word, that now we can do that with genes. That this new CRISPR technology is of much higher accuracy and efficiency than ever before possible, and that's why the game has changed. Okay. Intensive basic and preclinical research is clearly needed. Remember, this group could meet anywhere in the world and has gone all over the world since. Why did they start out on the Mall in Washington, D.C.? Well, that's where the money is. All right, keep sending the money. There's, from our tax dollars, tremendous amount of resources has gone into developing this technology, particularly because it's our most promising way of fighting cancer. And if you're writing a grant in federal DC to the National Institutes of Health or to other organizations seeking funding, if you can explain why your work helps us fight cancer, that's tremendous help in getting funding. So the scientists involved are saying, keep sending the money. We're putting it to good use. And we're also here in Washington, D.C., we don't want more regulation or more laws. We want to have the freedom to continue to develop this service. So that's why they're meeting in Washington, okay? And so there's a reminder that, and a desire to emphasize that we need to emphasize basic and preclinical research that's still very much needed. The proposed clinical uses are intended to affect only the individual who receives them. The technical term for this is somatic therapy. Soma is just the Greek word for body. So somatic therapy is you're just treating the particular patient that has presented to you. That there's substantial consensus that when you're just treating that patient in front of you, if there's some way through gene editing that you can help them, you should. I'll give you some concrete examples so this doesn't sound too abstract. The vector, the means by which you get the genetic correction into a person's body, is technically very difficult. And there's real progress being made in doing that in a more accessible and easy way. The very first ways we've been able to do it have been with cells that you can take out of the body, correct, and then put back. And the easiest cells to do that with are related to your blood. It's a very handy thing that you can take out blood cells, fix them, and then put them back, and they go where they belong. So one of the first examples of practicing CRISPR for human treatment has been in regard to sickle cell disease. There are about 300,000 people in the world today that have sickle cell disease. It's devastating. You have trouble processing oxygen. You have severe pain in your joints to the point of being hospitalized many times a year, and a life expectancy of about 50 years. So a devastating disease. Sickle cell disease is at root a genetic issue. You have 4 billion base pairs that record the information in your genes. In every nucleated cell of your body, you have a copy of these 4 billion cells, 4 billion base pairs in a particular sequence. That's what records the information. Sickle cell disease is just a place where one base pair out of 4 billion is flipped upside down. So instead of having a T, you have TA, right? Thymine adenosine. So it's a genetic defect which takes your normally plump red blood cells that carry oxygen and makes them concave so that they can't carry oxygen very well. They look like little C's under a microscope. And you can imagine with those two horns, as they go through narrow spots in the body, they get caught, they get stuck or clumped together. And that's why it's so painful and why it's so difficult to get enough oxygen, and you tend to be very anemic, okay, weakened, as well as shorter life. All right, with sickle cell, Victoria Gray was the first person we actually treated with CRISPR. As of October 23, she's been symptom-free now for 55 months from a one-time treatment. So by going directly into the genes that cause the disease, she's been delivered from the disease. Now since then, there have been a whole series of other patients who've been so treated, gradually ramping up the number of people, and so far, everyone who's been treated with gene editing for sickle cell disease has, from one treatment, been freed from the disease. Another example is beta thalassemia. This tends to be found in certain parts of the Eastern Mediterranean, and then wherever people have, ethnic groups have moved since. Beta thalassemia also is lethal over time unless you have very regular, large blood transfusions. There have been 15 people treated so far with gene editing for beta thalassemia. They no longer need blood transfusions and are leading normal lives. So we used to talk about gene editing as in the future distance. We're not at the point yet where you can get it at your local drugstore, but we are at the point where we actually have real human beings being treated with these methods, and for all intents and purposes, it appears that they've been cured. All right, so at this point, CRISPR is expensive, invasive, and at limited locations. But we seem to have a working treatment that is working for beta thalassemia, sickle cell disease, ADD, and it looks close for hemophilia, muscular dystrophy, Parkinson's, and a number of various cancers. All right. Okay, so that's somatic cell therapy. That's where you use gene editing to treat the particular patient who's in front of you. The next question that was addressed by the group working together was that of germline editing. And that's where you make a change in the gene that's inherited by that person's children, so that you're treating not just the initial patient in front of you, but also the children that they might have that come to live in the future. The consensus so far among the people who've been meeting regularly to talk about this has been that it's not clear enough, it's not proven enough for enough people to do things that will affect future generations. That so far, the intervention should just be somatic. So the way they put this was it would be irresponsible to proceed with any clinical use of germline editing until, and then they give a series of things, and at the end they say, as scientific knowledge advances and societal views evolve, the clinical use of germline editing should be revisited. So not now, until certain things have been established and there's a general social consensus that it's appropriate. So I did put it in gold there for you. What's that key word in the first sentence? Until, all right? The consensus of the people actually doing this work so far is not that there will never be germline editing. The current consensus is that it's not appropriate now. Okay. Maybe some of you have heard of Jinhua He, the Chinese geneticist who did germline editing without telling anybody. It's a really interesting situation. Right after he announced this at the Hong Kong conference, and instead of getting applause, there was general horror in the group. He disappeared. He was later surfaced in a Chinese prison, spent about 3 years as a ward of the Chinese state in a prison, and then they let him go because he had run out so far ahead of what his fellow geneticists had said was appropriate for that time. Because he'd gone right into germline engineering when the consensus was that we weren't ready to do that yet. So there's a lot more to that story, but that gives you that idea. Okay. The group then said what we need to have is an ongoing forum. We need to keep meeting to advise each other, to consult with each other, to talk with each other, to think about what's appropriate. And that this forum should be inclusive. That it gives a long list of people who should be involved in the discussion. Besides biomedical scientists, there should be social scientists, ethicists, healthcare providers, patients and their families, people with disabilities, policymakers, regulators, research funders, and I put this in gold for our talk today, faith leaders. That there should be a widespread discussion about what's possible and what we should do on into the future, because this affects all of us. So for the purposes of this lecture series and its reason for existence, let's talk a little bit more specifically about how different religions have looked at these questions. Now, first of all, I just want to emphasize that religion is not fading away, contrary to Durkheim, Weber, Marx, Freud. There's been a whole series of people who said that as we become more technologically able, as we get smarter and better educated, religion will disappear. It just actually hasn't happened. Okay? On the contrary, in 2015, Gallup did a poll in 163 nations, interviewing more than 1 million people. And here's what they found from that study. 81% globally claim to belong to an organized religious faith. Not fading away. 50% reported that they had attended a place of worship within the previous 7 days. Okay? So surveying over 1 million people randomly around the globe, 50% reported that they had attended a place of worship in the previous 7 days. Very specific. 56% affirmed that they believe that, quote, God is directly involved in things that happen in our world. Okay. All right, so if you're going to understand how the world works, you've got to understand what's going on in the world's religions. Now, religion is a very diverse term. Of course, it can refer to many different things. What do all of the religions have in common that we can call them religions? Edmund F. Perry and Walpola Rahula, a Buddhist monk, found one thing in common in their Cambridge University Press book called World Theology. You can be thinking in your own mind, what's that one thing they found that was true of Buddhists and Hindus and Shinto and Christians and Muslims? What was one thing that they all affirmed? That there is more to reality than the material. In other words, there are things that are truly real and that matter that we don't describe as material. Okay? That's something that seems to be clearly affirmed by all of the world's major religions. Now, from my own experience and study, I think there's one other thing that I've noticed that seems to be true of all the world's religions. So I would like to add that it seems that all of the world's religions think that it's a good thing to light candles. It's just so consistent for different reasons, for different occasions, but everybody thinks we should light candles. I'll just mention that to you. Okay, so what there is beyond the material, and for that matter, what the material is, varies among religions. And I'll just very quickly give you some examples, because this affects how different groups look at CRISPR technology. Judaism is about 13 million people in the world today. Judaism believes that the material is not God, but it is God's creation, to be cared for and to be repaired where there's been damage to it. The phrase that's often used for this is tikkun olam. So there's a foundational idea in the Jewish traditions, even within their variety, that the material is not God, but it is something that God has created, and if there's been damage to it, we have an obligation to do what we can to correct it. That sounds a lot like what people are trying to often do with CRISPR. And so there tends to be a certain amount of openness to that. The Latter-day Saints Utah, sometimes colloquially called the Mormons, they claim that they have about 16 million members globally. One of their most famous quotes is from one of their prophets who was president at the time, and that gives them great authority in the Latter-day Saints tradition, who said, "As man now is, so God once was. As God now is, so man may become." So who we refer to as God once lived like we do as human beings. And we as human beings can look forward that if by God's grace we progress sufficiently, we can be gods ourselves over particular communities and places. Boy, that's a tradition that emphasizes improvement and progress over time. And so the idea of CRISPR as yet another tool to physically improve our condition and place has a certain resonance. Among people in the Latter-day Saints who have tried to reflect on this, there tends to be a real interest in this as yet another tool for progress. Buddhism, about 500 million in the world today. Remember, for Buddhism, and there's so much more to this, but I'm just highlighting an aspect, the material world is in a significant sense a temporary illusion. And so if you want to alter your perception and experience of the temporary illusion, that's not that scary, it's just a temporary illusion. So there tends to be quite a bit of resonance and openness to changes there. Hinduism is about 1 billion people on our planet. Remember, Hinduism is dependent on an idea of reincarnation, that when you die, you come back either higher or lower, and that your main goal in life is to produce good karma and to suffer off your bad karma, and that you want to, over time, develop into a literally higher being. So in Hinduism, the idea of a transformation, reincarnation, of improvement of your status into a whole different kind of being is central. Islam, 1.5 billion people. The word Islam literally means one who submits to Allah. And there's no central teaching authority in Islam to say what Islam believes or says. There's no one official interpretation of the Quran. There's no one individual who has that authority. And so you have different imams in different centers of learning that have different judgments. There is agreement in Islam that ultimately it's God's will that one should submit to and that you should seek to carry out. Exactly what God's will is in regard to CRISPR, there is not at this time a consensus. All right, well, this leaves us then with the world's largest religious group. So what would that be? Okay, that's the Christian tradition in all of its variety. There are about 2.5 billion people in the world that describe themselves as Christian. Now, in our limited time together, I'm going to focus on this largest and most global of the world's religions, the Christian tradition. If this is your tradition that I will now describe, it might sound quite familiar. According to polls by Rice University's sociology professor Elaine Eklund, most scientists are theists, and many are Christians. And I have a reference there, Secularity and Science, What Scientists Around the World Really Think About Religion, Oxford University Press, 2019, if you want to see the statistical analysis to that effect. If the Christian tradition is not your tradition, you might use this opportunity to better understand many of your colleagues, clients, patients. It's the world's largest movement. If you want to understand the world and how it works, it's good to understand the Christian tradition. Or even you might gain some insights, as we often do, by exploring the perspectives of others. So I think as we talk in more detail about the Christian tradition, you might be able to pick up some insights or ideas, even if that's not your tradition. Okay, so let's do that. First of all, is the Christian tradition hostile to science? Absolutely not. We're talking about genetics in particular. Who was the first person to discover units of inheritance? The first person to describe human genetics and how it works? Pardon? A monk, yes, it was Gregor Mendel. Gregor Mendel discovered units of inheritance in the garden, in particular in the pea plants. He noticed yellow and green leaves and peas and how they interacted. He realized there was a mathematical model of how things were being inherited, and he described this in technical journals. He was the founder of what we now call genetics. What was his day job? He was a monk. So he spent every day in worship and in prayer and in Bible study, and each member of the monastery had to do something to support the physical life of the monastery. His job was taking care of the garden. He was a more efficient garden if he understood how plants process and how they crossbreed, and so he devised the study of genetics. So a Christian monk was the founder of the field. Another example would be, we could easily argue that one of the most influential people alive today in genetics is Francis Collins. Francis Collins and his team discovered the gene for cystic fibrosis, the most common genetic condition in the United States. He went on to be the director of the International Human Genome Project, which brought labs together all around the world to make the first working copy of a human genome. So he led that effort. And he then went on to be appointed by the President of the United States to be the director of the National Institutes of Health, the world's largest funding of research in medicine, and did such a good job that the next president reappointed him, that had never happened before, and did such a good job, the next president reappointed him. So he's served as director of NIH longer than anyone else in the history of the United States. Francis Collins is very open about his Christian faith. He had a best-selling book a few years ago called The Language of God, published by Harper One, and it's an interesting read, both about the Human Genome Project and about also how, as a medical student, he became a Christian. He grew up in the Shenandoah Valley near Stanton, Virginia, and not in a Christian home or family, but came to faith as a medical student and continues to be very open and clear about his Christian convictions, as well as being one of the most famous geneticists in the world today. So is the Christian tradition hostile to science? Well, it's actually amazing how many Christians have been instrumental in genetics. Let's broaden that out to wider sciences in general. The American Scientific Affiliation, if you want to look this up on the web, it's just under asa3.org. The members affirm the early summary of Christian faith called the Apostles' Creed and seek to bring that tradition into dialogue with the best of the sciences. And so just real quickly, some examples of members of the ASA: Francis Collins, that we just mentioned, Jennifer Wiseman, who spoke in this series not too long ago. She's the senior project scientist for the Hubble Space Telescope. Rosalind Picard, the founder of Effective Computing at MIT. Ian Hutchinson, Nuclear Physics, Princeton University. Owen Gingerich, Astronomy, Harvard University. Santa Ono, the president of the University of Michigan. David Vosberg, Chemistry at the Claremont Colleges. I should certainly mention Phil Smith, New Bern, North Carolina. All right. The ASA has for 75 years published a journal called Perspectives on Science and Christian Faith, which has academic peer-reviewed articles talking about the full range of the best of the sciences and how this might relate to the Christian tradition. I brought along for your interest and use some back issues, copies which you're welcome to take as a sample, free. And you can even take more than one. Each issue will have entirely different science topics that's being integrated in this conversation. So feel free to paw through and see if there's a particular issue that has a topic of interest to you, and you're welcome to take a look at that. Okay. So that's on the table up here in the front if you're interested. There are other academic peer-reviewed journals pursuing the dialogue between science and Christian faith. I think of Science and Christian Belief, which comes out of London, Theology and Science, which is published out of the University of California, Berkeley, Zygon, which used to be out of Chicago and now is out of Amsterdam. There's a whole world here of discussion between people who are trying to understand the connection between the Christian tradition and the best of the sciences. Okay. So if you're trying to do this dialogue, you're going to look for 4 primary sources in the Christian tradition. All of the various Christian traditions use these 4 primary sources, just different ones emphasize one more than another. There, of course, is experience of God at work in one's life, Scripture, tradition, what has God taught or explained before, and reason. God gave you a brain, you're supposed to use it. So Presbyterians will tend to emphasize one of these more than the other three. Roman Catholics emphasize one more than the other three, but all four of these are present in all of the major Christian traditions. So let's use those tools and just talk briefly about how the Christian tradition might view the use of CRISPR and how to use it properly. Okay, first thing to remember is that nature is constantly changing. How many of you recognize this particular volcano? It's Mount St. Helens. Well done. Okay. At 8:32 a.m. Pacific Standard Time, May 18th, 1980, this 10,000-foot mountain exploded. 425,000 tons of rock and ash roared east parallel to the ground at 425 miles an hour. Within 15 minutes, 54,000 acres of pine trees were open plain. A mile-long lake appeared, and a river changed its course. There's a picture after the explosion. Here's some of the timber that was laid down. You probably don't realize the scale. Do you see the people in the picture? I'll give you a red arrow. There they are. If that gives you more of a sense of the kind of timber that was laid down in just a few minutes by the blast. Nature, by nature, is constantly changing. Now, it's not many of us as human beings that live in a place where we can just live off the land without changing nature to survive. In other words, the minute you sharpen a stick to spear a fish and then you hang it over flames to cook it, to eat, or maybe bend a couple palm fronds together to sleep under at night or to fend off the rain, you are by nature changing the environment that you are living in. Human beings are not so much like vultures that can just watch for something to die and scoop down and eat it. We're more like the part of nature that we call beavers, that intentionally change our environment in order to survive in our environment. It is our nature to change nature. Now, I don't say that to be anthropocentric. I'm not saying that to say that the world exists just for us. From the perspective of the Christian tradition, I'm trying to be theocentric. That it's not just about us, it's about God and what God is doing. Before I came south, I was at McMaster University, what many people would claim is one of the leading research universities in Canada, located outside of Toronto. And the theme of McMaster University, the slogan that's in the seal, is Ta Panta en Christoi Synesteken. That could be roughly translated as all things hold together in Christ. It's Jesus Christ that makes sense of the world. By this perspective, the world is not God, but this is God's world. So maybe you know the famous text, "For God so loved the world that he gave his only Son, that whosoever believes in him should not perish, but rather receive eternal life." That's from a book about Jesus by somebody named John, the 3rd chapter, the 16th sentence. For God so loved the world. The Greek text is, for God so loved hakosmos. It doesn't say for God so loved men, or for God so loved women, or for God so loved human beings, anthropoi. It says, for God so loved hakosmos, the whole shooting match, the whole creation that God had made. That God cares about all of creation. That human beings were created uniquely in God's image, that they have a special place in the overall creation. That human beings have a unique capacity, an ability to know God and to know that they know God. That human beings like Monet, there's no other creature. You can give a paintbrush to an elephant and they can make something abstract, I know that. But there's no other animal besides human beings that can paint like a Monet. There's a level, a capacity in human beings that's distinctive. And along with that comes a relationship with God that's distinctive, and a calling from God that's distinctive. That human beings are put in a place where they're responsible for how they care for and take care of the world that God has made. So Genesis, that just means beginnings, the first book in the Hebrew Bible and in the Christian Bible, says that God placed human beings in a garden, not a wilderness. A garden requires choices. You have to decide, are you going to encourage the roses or the aphids? But you can't maximally encourage both. If you have a wilderness, you just pass through. If you have a garden, you're responsible for how it develops. You make choices that make a difference. It's still God's garden. The Genesis chapter 2, sentence 15 mandate is to till and to keep the garden. That word to till the garden is the same word that's used when it said, "As for me and my house, we will serve the Lord." It's not till as in rend or cut up or destroy. It's till as in serve. And that word keep is the same word that's used, "May the good Lord bless and keep you." That there's a responsibility to care for the world that still belongs to God. So from this perspective, as we care for the garden, what is does not automatically tell us what should be. We're to care for and develop ourselves and the rest of creation, like in a garden. You tend it in a particular direction to help it to thrive, to reach its full potential. In the Christian tradition, the word Christ, Jesus of Nazareth, the Christ, is at the center. Jesus of Nazareth is described as spending the first 30 years of his life doing what? Big hint, right? Is a carpenter, a teknon, a builder, somebody who shaped stone and wood to better serve people. And in fact, Jesus taught his disciples to multiply their resources to better serve God and people. After all, that's what really lasts. If you want your life to be invested in something that's going to last, it's God and people that last. So Jesus talks in Matthew chapter 25 about taking your talents, whether that's money or ability, and multiplying it. And then his disciples asked, well, then how do we invest it? Where should we spend it? To what purpose and in what way should we develop our entrusted resources? And Jesus says you should give water to the thirsty and food to the hungry. You should clothe the naked. You should visit those in prison. You should care for those who are sick. That you should multiply your resources and use them to better care for God and for people. If you really believed that, would you build a beautiful brick building like where we are now? Oh, absolutely. If you built it to help people to gather, to know and worship God, and to better love one another. So in Matthew chapter 25, Jesus praises those who were prepared for the long haul, who multiplied entrusted resources to care for their neighbors. That's a motivator for science to understand our world and for the development of technology to shape it, exemplified in Jesus, who is described as spending his first 30 years as a builder and his last years of public ministry as a healer. There's approbation for those who did not do what they could, who did not care for their neighbors when they could have helped. So what are we called to do from this perspective, from this tradition? I would say three things. We're to sustain the world that we've received. We're to restore it where there's been damage. And we're to improve it where we can. Would that include even our bodies? Well, what was the sign mandated to Abraham to show that he and his descendants were the people of God? Here's a big hint. Okay. That's an Ishmael, a ceremonial knife. used to cut off a normal part of a male human body, modifying a human body to show that somebody belonged to God and wanted to serve and follow God, as a sign given by God. This is my covenant with you and your descendants after you. Every male among you shall be circumcised. Genesis 17:10. What could be more central? This is a central direction to modify the human body to show that one is part of God's people. Now, if you ignore your teeth, they'll go away. And my mother would want me to go to great pains to emphasize that although she's the one who told me that, this is not a picture of her. All right? But it's certainly an example that if you ignore your teeth, they will go away. What are we supposed to do with our teeth? We should sustain them. How do we sustain our teeth? Well, you brush them, for example. How should you restore your teeth? Well, if you get a dental carie, a cavity, you should have it cleaned out and filled. Fill your cavities. And is there anything we can do to improve our teeth? I am thrilled that my daughters have never had a cavity in their life. Part of the reason for that is because we gave them, when they were quite young, just had their adult teeth come in, a fluoride treatment that actually strengthened their natural tooth enamel so that they do sustain their teeth, they take care of them, but they've never had to restore them because their enamel is actually even stronger than it was naturally, and they've never had a cavity. I'd argue that was appropriate. Sustain your teeth, restore them if need be, and improve where you can. You've probably never seen somebody who looks like this. But in parts of the world you still do. Well, not really. I should take that back. It used to be that millions of people died of this every year on a global basis. That's smallpox. Okay? And in fact, I can probably guess that everybody in this room over the age of 50 has a circular scar right up here. And anybody in this room under 50 doesn't. And the reason for that was the development of a vaccine against smallpox, where we spent years doing it, but we tracked down all the live cases of smallpox in the world until there was no more wild smallpox. There was nobody with smallpox to pass it on. It died out. We actually eliminated the species. There are actually only 2 cities in the world that still have viable live smallpox: Moscow and Atlanta. Okay. Of course, why do Moscow and Atlanta still have smallpox? It's the Centers for Disease Control in Atlanta and its equivalent in Moscow have smallpox in deep freeze so that if somehow anything ever gets out again, they can quickly develop a response. But we wiped it out as a global phenomenon. I would argue that that's a fearsome thing to wipe out a species, that we shouldn't do that lightly, but that was justified and appropriate to wipe out something that was killing millions of human beings a year. That would be gardening, that would be appropriate improvement. A biblical basis for this. I think, for example, of the prophet who lived 2,500 years ago, Micah, again in the Hebrew Bible and the Christian Bible. What is it that God requires of you? God requires that you seek justice, love kindness, and walk humbly with God. It's really interesting that in global bioethics, we have 4 key questions or principles that you hear as much in Mahari Medical School in Chiang Mai, Thailand, as you do in Toronto at McMaster University's medical school. And that is that you should always ask, are you doing what is beneficent, produces good for your patient? Are you avoiding harming your patient, non-maleficence? Are you giving your patient more choice by what you're doing? And is this the best use of always limited resources? Justice. And these actually line up with what the prophet Micah said 2,500 years ago. Seek justice, justice fairness, love kindness, beneficence, non-maleficence, walk humbly with God, show respect for the autonomy of and decisions of other people. So, applying those, let's be really concrete. I would suggest to you, from my perspective, I think trying to think of this Christianly, but I think in a way that's resonant for many other traditions and perspectives, that if you're asking if something should be done with CRISPR, the question's not so much somatic germline or cure, enhance. I think you should ask these 4 questions. So I'm making a concrete proposal. I think you should ask, number 1, is the intervention you're doing safe for everyone involved? At least don't make the patient worse. Okay? Non-maleficence, don't harm. So number 1, is it safe for the recipient and the people who are doing the process as well? Number 2, is the effect that you're pursuing a genuine improvement? Is it a genuine improvement? Before you start expanding human lifespan, be confident that it's a genuine improvement to expand human lifespan. I mean, whatever it is you're pursuing, is it a genuine improvement? That's beneficence. Number 3, does it increase the recipient's choices? People often worry that if you intervene genetically, you are taking away people's future choices, that you're predestining them. But if you make genetic choices that actually give them more choices, that's not predestining, that's honoring their opportunities. If you help a child learn to read, you have not taken away their choices, you've opened up whole worlds of information and experience that they didn't have before. Okay? So you only do interventions which increase choices, not decrease them. And then 4th, is this the best use of always finite resources? We just can't do everything for everybody. You have to think about the larger picture. If you make this intervention, how is it affecting other people and circumstances? And is it the best use of always finite resources? I will argue that if you're looking at a particular question of using CRISPR, and it answers those 4 questions well, it might then be an appropriate intervention for the presenting patient, and through them, maybe even for their children. Now, we've got to deal with the elephant in the room. Of course, we have the great concern about eugenics. Eugenics literally just means a good birth, trying to give people a good physical start in life. But the eugenics science was commandeered socially between 1920 and 1950 by people who overpromised what genes cause, neglecting the formative influence of social experience and individual choice, and in some horrific cases used the pursuit of genetic health as a weapon against, rather than a tool to help, certain groups and individuals. So I'm thinking, for example, of Nazism, which in a very gradual process claimed to just be seeking good health and actually spun into a racist program that murdered untold millions. Neither of those abuses is endemic to gene editing. Long before the possibility of gene editing, Chevre Dor Yeshorim, a Jewish group, began successfully working to free Ashkenazi Jews, an ethnic group, from losing their children to Tay-Sachs disease. Chevre does not attack the group or individuals afflicted with Tay-Sachs. It works to free their group from Tay-Sachs. Tay-Sachs was a condition that if you inherited it from mom and from dad, the child looks perfectly normal, but at about 6 months of age goes blind and then loses what limited mental capacity it has and then dies. So a devastating, horrifying genetic disease. What Chevre Dor Yeshorim did, in the name of a good birth, giving people the best start they could, they said, we're going to test everybody who's interested, and we'll keep it secret what your test results are.
But if you get romantically inclined, if you think you might want to have children with somebody, then you can call us and you can give us your code number and their code number, and we'll tell you either that there's no chance of you giving Tay-Sachs to your child, or we'll tell you that there's a 1 in 4 chance that you will give that disease to your child. or that it's likely that one of you is a carrier and the other one is not, so it's 50-50 if you pass on the tendency but not the disease. They found that when people had this information, some couples decided, boy, that's so important to us to have biological children, we're just not going to get married.
Some people said, we want to get married, but we're going to adopt. Some people said, we'll get married, but we'll use artificial insemination by donor so that we can guarantee that the sperm that's involved does not carry the disease. Some said, we'll just take the chance. We'll just, even with this information, we'll continue. People had very different responses. But the number of children born in that community with Tay-Sachs disease is now about 1 or 2% of what it was before. So people have taken actions and choices that have dramatically limited the number of people harmed by Tay-Sachs disease. Now that was a sociological response, not a gene editing response, but it gives an example that you can be concerned about the genetic heritage you give to your children without it being a racist attack like the Nazis. Okay, so I'll suggest that changing our received nature could never offer us salvation or utopia. But we can and should tend the garden and help our neighbors. Now, you probably know, a gathering of cows is a herd, of whales is a pod. What's a gathering of crows? Murder. A murder. Well done. Again, this is an erudite group. Very impressive. A gathering of geese. Gaggle. Gathering of lions. Pride. Gathering of ethicists. It's not official, but I really think we should call a gathering of ethicists a worry. I think that would be a worry of ethicists. That just fits. Okay, I want to suggest that ethics serves an important purpose when it warns us of the danger of pride, but it should also warn us of the danger of apathy. It tells us what not to do, but it should also remind and encourage us as to what we should do. Christian ethics is neither lawless nor legalist. It's about being set free through and in Christ to live as God created us to live. And that's going to entail things that we shouldn't do, but also things that we should. It's about being set free to live life as intended. Now, if you want to hear more about this, I can vouch for this particular book. It makes a lot of sense to me. And so I'll just mention it to you that this exists. But boy, that's a lot that we've covered very quickly. So we'll make this the end of the initial presentation. And I believe, how do you want to do this? You want to give a certain amount of time for people to take a break? And so how soon should people come back?
5 minutes.
Okay, in 5 minutes, we'll take questions. And I believe you're writing out question cards. You can hand those in. And your questions will drive how we discuss things next.
So again, many thanks for coming, and Jim, especially thanks to you for coming. It's been really special. Many of you had questions about gene editing. Is it time now to have no more sickle cell disease? Or why does it cost so much at this point? And how much did it cost? What about gene editing for other diseases like Parkinson's disease that you made mention of? And are we going to, that's a good place to start, I think.
Okay. All right, very thoughtful questions. Anytime you develop a new therapy, it's usually very difficult to do it the first few times. And you usually want to start with a very small sample of people in case there's some untoward effect that you weren't expecting. So that's why I gave you the example of with sickle cell disease, starting with the very first patient and seeing how it went. When it went so well with her, then it expanded to, say, 10 people, and then it goes to another 15, and you keep increasing the numbers. If there's something that only goes wrong for 1 in 100 people, you don't know that until you've tested it on about 100 people. So there's a gradual process of ramping up when a new treatment becomes available before you go global, before you spread to many, many people. So that incurs a much more narrow locus of where a particular treatment is available and how soon it's available. Usually you live near a particular research center where the studies are being done if you're going to be eligible to be in an early research study. So there is a limitation on location. As far as cost goes, like anything, the very first automobile that was made was extremely expensive, like a Rolls-Royce, handmade. But then when Henry Ford developed a method of mass production, the cost per unit came down dramatically. Something like that happens with most medical treatments. They tend to be very expensive at first, and then we try to develop ways to make it more widely available if it's clear that it's being helpful to people. So, for example, that first patient who was treated for sickle cell disease, all told, when you take all of the laboratories that were involved and all of the special chemicals that were devised for that very purpose and people figuring things out for the first time, I think the current estimate is that about $1.5 million to $2 million has been spent on that first patient. So prohibitively expensive. But our history is that once you find something that works, it's usually possible to start finding ways to economize and to start dramatically bringing the price down. I'll give you an example specifically in genetics. The Human Genome Project spent hundreds of millions of dollars to sequence one person's genes the first time. And it was announced from the White House when it was completed. I mean, it was an amazing achievement. We can now read all of the genes and all of the base pairs in your particular human genome for about $200. Okay, so from hundreds of millions to $200. So that's the kind of process that we're following. Now, in fact, there are more and more physicians that are starting to are starting to call for a complete reading of your human genome as part of an annual physical. The problem is that it's so complicated to interpret. And the barrier is having people with the expertise and with the know-how to explain what it means. So you can get the test done, but the real effort is in figuring out how it applies, what it means, what do we do next. Right? So I gave you these examples of some of the treatments that are starting to happen because it used to be theoretical. We used to say, we hope that someday this might help a patient. We think it looks like it will help a patient. We're actually at the point now where the numbers are small, but we have human beings who've been cured. And now it's a gradual process and a very important process of making it more economical. And then as it becomes assured that it's efficient and it works, making it available to more and more people. So I don't want to give false hope or talk about a particular disease. If you want treatment for it in the next 6 months or a year or 2 years, it's just going to take a while to roll this out. But it's amazing how this will affect the next generation and start to affect our current generation. It actually can be done, and we're learning how to do it safely and effectively and efficiently. All right, maybe that's where to go start.
This may be the definition of delayed gratification. There were particular questions about Parkinson's disease and pulmonary fibrosis and whether those things might be able to be changed by gene editing. Yes?
Okay, so specifically, an example of something like Parkinson's. I'm very aware of that. I have a father who is stricken with Parkinson's. If that's the particular condition that you're facing, talk with your specialist who's treating you and talk with them about what kinds of treatments are available. And if your specialist is aware of something related to CRISPR that might be helpful, it'll probably be connected to a particular academic center where people have focused on that research and where they do quite possibly have small numbers of people who are starting to be tested for that and treated for that. And then it becomes a question of, well, if they're doing this at Northwestern University in Chicago? Am I willing to move to Chicago or to travel there regularly? Your particular specialist, or even starting out by going to the National Institutes of Health website, you can search for what studies are being done. And you might want to consider participating in something that's a very early stage. But yes, it's going to take time because we want to protect people from any harm in the process. It will take time to have any kind of regular treatment for many of the conditions that it will eventually probably be quite treatable. But it's going to take time. In the meantime, you can ask your physician about if there are particular studies that you might want to participate in. So an in-between answer on that too.
Thanks. There are several questions that I'm going to kind of group together about the negative aspects of gene editing. For example, is there the possibility of engineering a Sparta state of warriors? Would Putin do that, for example? Or what's the likelihood that geneticists are going to follow their guidelines? Maybe we already have an example where one did not. Yeah. Okay, sure. Yeah.
Another one? Okay. All right, that was a bunch of questions already. Okay. As far as engineering warriors or the super soldier, it's an incredibly slow process. If you want a super army through genetic means, it's going to take 20 years, and most people aren't patient or willing to wait for an armament that's not going to help for 20 years. Also, I don't think super soldiers would make it through my 4 key questions, where you're asking, is this harmful? Is this a genuine benefit for the recipient? Is this increasing their choices? Well, no, you're actually destining them for a particular use. Is it the best use of limited, finite resources? Well, no, there are many other things we could be doing that would be better. So I would argue for ethical reasons that's not something that one should pursue. Now, can you have a rogue nation try to pursue something like that over time? It's possible. If you're talking about what the individual geneticists do, science today is collaborative. Nobody has the knowledge base to make advances in science by themselves. And so scientists have learned that it's extremely important to cooperate with others and to have a good reputation for cooperating. So that's why the example that, the exception that proves the rule, here's one geneticist, Zhang Kuihe, trained at Stanford, goes back to China. He does something that his fellow geneticists say we should not be doing it at this point, and he's out of the discipline. I mean, he's ostracized. He's lost his ability to cooperate and to be part of the science of genetics because he went off and did something on his own that the others said, no, we shouldn't be doing that at this point. So there is a very strong, and that would be an example application, feedback loop when somebody does something that the rest of their professional colleagues thinks is inappropriate. They lose the cooperation that they need to proceed. So those were 2 key questions, and that would be an initial response to each.
So there are 2 questions about lifespan, life expectancy. Are we now able to genetically alter lifespan? And will we at some point be able to extend it indefinitely and maybe eliminate death?
Well, you all realize that if you walk around a graveyard here in Newbury, that you will see some people who lived to 70, 72. Every once in a while you might see an 86. But really most people were dying at a much younger age. Are you less human because you're living longer than most people used to? Is that an improvement that we now tend to live much longer than people used to? So this question of is it appropriate to extend the human lifespan is one that we're already living through, a grand experiment and practice is something that we're doing. So that's not new just with genetics. Now, are genetics involved in lifespan? Definitely. You're extremely influenced by your environment. I mean, you get hit by a truck and die at 19. I think of Sergei Grinkov, a Russian athlete in superb condition, who was very successful in the Olympics and died at the age of 22 from a heart condition. And I compare him to Winston Churchill, who smoked copiously, drank alcohol constantly, and lived to what? Anybody know the age? He made it into his late 80s at least. I don't remember exactly. Does your choices makes a big difference in your lifespan, but your genes really do. And we do know genetically there are these telomeres, these endings on your genes, that as you age, you lose telomeres. And so there's an interesting question that if you can restore telomeres with gene editing, will that cause people to live longer generally or improve their odds? There are things like that that look promising. But your age is so tied to a whole series of other complex factors, it's not going to be just a genetic approach, if indeed we are able to expand lifespan. Yeah.
So the answer to that is to eat less and exercise more. So has there been or is there a role for artificial intelligence or machine learning in genetics?
Yeah, already. Is there a place for artificial intelligence in genomics? Amazingly already. Your genes encode for particular proteins. That's a structured chemical which the way it links up with other structured proteins or chemicals is what forms your body and what informs the processes of your body. So these shaped entities that we call proteins are central to health and to how your body works. It used to be, if you wanted to know what was the shape of a protein, there were people who earned PhD theses for figuring out over 20 years or over 10 years or 5 years, one protein. That's a computational issue that lends itself to the kind of intelligence that artificial intelligence is good at. And so artificial intelligence has already been used to create, you might call them encyclopedias of if you have this genetic recipe, this is the protein you will get. And what used to take decades to carefully figure out, artificial intelligence can churn these out just in a few seconds. And so now we have libraries of protein structures, hundreds of thousands of them, that we can then apply to see which one's happening in a particular case, or if we put this chemical in, or if we change this, that will happen. We have whole libraries of proteins that have been described for us by artificial intelligence that just simply weren't available before. So artificial intelligence is very good at certain high computation tasks. It does not function like human being general intelligence, but it's very good at what it does. And that's why AI can be really good at chess or Jeopardy or Go. These are very delineated tasks that are primarily computational. And so they're very good at that. When AI gets into medicine, if it's highly computational, it can do it for us. If it's general intelligence, really the best thing it can do is serve us as an advisor, not to replace human intelligence, but to augment human intelligence. So an example of where this can be very useful is when a physician is considering, from what I see in your symptoms, I think this particular drug would be most helpful to you. And then type that in to the medical log, and then have an artificial intelligence say, do you realize that this is going to have this contraindication with this other drug the patient was prescribed 2 years ago? That's computational. That's where you can look statistically and you can see, and a computer's very good at looking at that long history and the other 50 drugs a person's taken and saying, are you aware that there might be this bad interaction? And then it's a judgmental question for the physician about whether it's worth that risk, if that risk is likely in this particular patient. But bringing it to the physician's attention is a very useful consult. So computational, yes. Final general judgment, no.
So the answer about for medicine and diagnostic and treatment purposes is probably within a few years, any artificial intelligence program can do what I did for 30 some years, and probably better. But you still need a relationship with your physician for just what he was talking about, to think about risks, benefits, how it fits into your particular set of circumstances. So don't give up on us doctors yet. So we have one very specific genetic question and then a broader question about ethics. But could you define genotype and phenotype?
Sure, yeah. No, I'm glad, if that's not clear, that's really good somebody asked that. Genotype is what are the particular base pairs of DNA do you have in your genes? So that's a description of, remember you have those 4 billion base pairs that give the recipe for all of your body structure and function. How does your body know how to make an eyeball or how to make a brain or how to make blood? The recipes are all there in your genes. That's genotype. Phenotype is what's the particular tissue or body you ended up with. So for example, identical twins, what's identical is their genes. Identical twins have the same genes, but they'll have different fingerprints. So that tells you that their genotype is the same. Their phenotype has a significant difference. Phenotype, what you actually end up with, is influenced by a whole series of other factors. So genotype, your genes; phenotype, the body you actually have. I think that covers it.
Precisely. Various forms of penetrance is another word people talk about, how much a gene is expressed in a given individual. So on the ethical side of things, how are we going to be able to provide these expensive, so far limited treatments to a broad variety of people, to a diverse patient population? And you may have already addressed this, but the questioner says that, as you mentioned, Jesus told us to help the sick. Does that not make us obligated to use these treatments? We shouldn't question whether we should use them. We were obligated to use them.
You'll notice that those 4 questions or principles that I mentioned, technically in ethics we call them prima facie. That means that they're a genuine obligation, but that they're not an absolute. And in fact, they will often conflict. So for example, when we do open heart surgery, why do we call it cracking the chest? Well, it's because you're sawing the sternum that's designed to protect the heart underneath, and then you use retractors to pull the ribs apart, and you get a whole lot of cracking and popping. Open heart surgery is really hard on the patient. So how can that fulfill the standard of non-maleficence? Don't make your patient worse. Don't harm your patient. You're making them a lot worse. You're really harming them. But we say that it's not an absolute, don't harm your patient. We're saying it's an obligation, don't harm your patient. But it can be overridden by a higher moral concern, saving their life. And actually, in the technical literature, we would say that there are then 4 standards or provisions: that you do it as minimally as possible, that you make amends for having done it, and that it was a last resort. And that's why your physician tends to say, before they do open heart surgery, they say you need to change your diet or you need to get exercise. They try to do a bunch of stuff first before they do open heart surgery, because they know open heart surgery has a lot of damage involved. It might be worth it, but it has a lot of damage. Okay? So those 4 principles are genuine obligations, but they can be overridden by a higher moral concern under certain circumstances. So when you say that we have an obligation to help someone genetically, you might very well be able to make that case that you should do gene editing to help that patient who can be helped by gene editing. But you still have to answer the question not just of beneficence, you have to answer the question of non-maleficence, what harm are you doing? You have to answer the question of, are you increasing their choices or decreasing them? And you have to ask, is this the best use of always limited resources? So you can say that there's a genuine obligation, but that doesn't settle it by itself. You then have a further analysis to make sure you're really considering everything that needs to be considered before you proceed.
Jim, Dr. Peterson, many thanks again. This has been really a really eye-opening, illustrative presentation. We've learned a lot. And to the audience, thank you for your questions, very thoughtful, probing questions that really allowed Dr. Peterson to clarify some of the things he said in his talk. So it's really been just great. Thank you very much. And we're delighted that you could make it here. Thank you.